Students, good evening. I welcome you all again for the biology detailed solutions mock test 6, okay, for part 1. So let us start the discussion. Okay, so let us start the discussion of this biology detailed solutions. Okay, so it is going to be very interesting today. Yes, just 1 minute. I'll just open up the app. So we are going to start the the question and answer solution. Let us go to the first question. Okay, the period. Okay, the period from birth to natural death of an organism. Okay, it represents. The question and answer solution. Let us go to the first question. So it represents lifespan, life cycle, natality and mortality. So what is the period, okay, that from birth to natural death of an organism that it can represent? So when you look into it, like when you take the lifespan, yes, you are right, Srivatsan, the answer is the lifespan, because as you see the natality, what do you mean by natality? It is the number of birth that is during a given period. What about the mortality? It is the number of death in a given period. And we all know the life cycle, right? Okay, it is from a series of changes that is going to take place in the life of the organism, which includes its reproduction also. So what is the answer? So the period from birth to the natural death of an organism. So it refers the lifespan. So we can see because of the scientific advancements, the lifespan of the human has increased. Yes, do you all accept with that? So it is the lifespan. So because right from birth to the natural death. Okay, so that is the lifespan. So you can see each and every organism can live only for a certain period of time. Yes, every organism has its own lifespan. So the period from birth to the natural death of an organism, it represents its lifespan. So what do you mean by natality? It is the number of birth during a given period, which is in the population that are added to the initial density. And what about the mortality? It refers to the number of death in the population during a given period. So the life cycle is the series of changes in the life of an organism, including the reproduction. Okay, so that you need to know. So the exact answer is what? It is the lifespan. Okay. Which cells are stimulated by the FSH in males? FSH is follicle stimulating hormone in males. Sertoli cells, spermatogonia, interstitial cells of Leydig, cells of the ejaculatory duct. Now when you talk about the FSH, like this follicle stimulating hormone, it has got a role to play with spermatogenesis. Like it is not going to be direct, rather it is going to act. Okay, it is going to act how? It is going to act indirectly. Okay, it is going to act indirectly through the Sertoli cells, and it is going to bring about, okay, it is going to bring about the spermiogenesis. The spermiogenesis is the process where the spermatids gets changed it into sperms. So you can see the FSH, right? What is happening there? It acts on the Sertoli cells and it stimulates the secretion of some factors which helps in the process of spermiogenesis. So here the spermatids are transformed into spermatozoa. It is called the spermiogenesis. So we have the entire sequence, that is the process of formation and maturation. Okay, the process of formation and maturation of sperm, you call it as what? The spermatogenesis. But spermiogenesis is the transformation of the spermatid to a mature sperm. So that is spermiogenesis. And here Neelambari, Srivatsa, 1 minute, understand, like you have given the answer, interstitial cells of Leydig. Actually interstitial cells of Leydig, they are acted upon by luteinizing hormone, not FSH. So LH acts on the Leydig cells and it stimulates the synthesis and secretion of androgens. What is androgen here? The main androgen in men, it is the testosterone. So androgens in turn stimulate the process of spermatogenesis. So Srivatsav, Neelambari, you are able to follow. So interstitial cell, it is the luteinizing hormone. And what about the FSH? It is the Sertoli cell. Rather, it is going to help in the indirect way of performing the, what is that? The spermiogenesis. So it has got a role in spermatogenesis. Okay, very good. Okay, we'll move to the next question. The answer is Sertoli cells. A functional mammary gland is characteristic of all female mammals. The glandular tissue of each breast is divided into dash, that is A option, containing clusters of cells called alveoli. The cells of alveoli secrete milk which is stored in the cavity, that is the lumens of alveoli. The alveoli open into B dash. The B option of each lobe join to form a dash, that is the C option, and then several dash, that is option C, join to form a wider dash, option D, which is connected to dash, that is the option E, through which milk is sucked out. So here you have to know the entire anatomy of the mammary gland. Okay, so I hope, let me explain you all. So as you all see here, the mammary gland, it is actually a modified sweat gland. So here what is happening here? What happens? It is rudimentary in males. Because of the hormonal influences, it gets developed in the case of the female. Now what is happening here? You can see the glandular tissues, like they will be divided here into 15 to 20. Okay, they will be divided into 50 to 20 mammary lobes. Okay, and what is going to happen here? They contain a cluster of cells which we call it as nothing but the alveoli. Now the cells of the alveoli is the one which is going to secrete. Okay, it is the one that is going to secrete what you call the milk which is stored in the cavity. Okay, so they will be stored up in the cavity, that is the lumen, and they'll all will join and open up into the mammary tubules. The mammary tubules in turn will join to form the mammary duct, and then the mammary duct in turn, okay, they form what is called the wide region, the mammary ampulla, and all this mammary ampulla that in turn which will be connected by the lactiferous duct through which it is going to be sucked out. Yes, Neelambari, Srivatsav, Tasmeen, Lakshmi, all of you, please post your answer. So I just explained you the structure of the mammary gland. Now you see lot of options they have given. So which should be the right option? I already said it is 15 to 20. But here there are lot of tubules that will join to form the mammary duct. The mammary duct in turn will join to form the ampulla. You can see here, and all that open up what is the lactiferous duct. Yes, very good Srivatsav. The answer is B option. So here it is 15 to 20. So the glandular tissue of each breast is divided into 15 to 20 containing clusters of cells called alveoli. The cells of the alveoli secrete milk which is stored in the cavities of the alveoli. The alveoli open into the mammary tubules. So they open up where? Into the mammary tubules, and the mammary tubules of each lobe will join to form what is called the mammary ampulla. Okay, the mammary ampulla. Then the several of the mammary ampulla will join to form the wider, okay, that so it is the duct here, okay, and this the wider part is always, remember, it is the ampulla. Now all the ampulla will join and it forms what is called the lactiferous duct. Okay children, you are able to follow? Yes, excellent Srivatsa, you have given the right answer. Yes, very good. We'll go to the next question. Which of the contraceptive method cannot be practically used? So which of the contraceptive method that cannot be practically used? Condom, copper releasing IUDs, implants, lactational amenorrhea. Now when you look into this, condoms, they are widely used. What about IUD? Like I said, that Lippes loop or Marina loop, which is now recently, that is the one exported, what is called, which has been imported, and we are following that IUD in practice now. It's been fit up and used for 5 years altogether. So it is not only having copper, it's not a copper containing, but it has got the hormones. So they are released in very minute quantity that prevents the implantation. So that way we are using. So the implants, like in terms of derma patches, so much have come up. And then lactational amenorrhea. So this is what we will just see. Amenorrhea means anything in front. In our medical terminology, we put means absence. Okay, so there is absence of menstrual period. Why should this happen? This is because one is the pregnancy. Okay, that is normal physiological. But why it should happen? Yes, exactly, Neelambari, you have given the right answer. Yes, lactational amenorrhea. It is considered to be one of the traditional and natural method of contraceptive method, but it is not a safer method. Let me explain you what is happening. So what is happening here in lactational amenorrhea? See, when the mother after her delivery, she will start feeding up the baby. So what will happen to her prolactin levels? Definitely the prolactin level which is secreted by the anterior pituitary gland will be more, because what is happening? The baby is being breastfed, and definitely prolactin is a lactopoietic hormone, so it is going to be increased. So what is going to happen? Because of this, there is going to be the negative feedback inhibition to FSH and LH, where these hormones, which we collectively call as the gonadotrophins, they are not released. They are not released. So what is going to happen? There will be no follicular development nor ovulation. Yes, so the women will have absence of her menstrual period during the breastfeeding time, which we call it as lactational amenorrhea. So they say that if the mother is going to have a sex, the intercourse, she may not get pregnant, but this cannot be taken as a chance because it is not a safer method. So many chances of failure, and it is not even a reliable method. So that you need to understand. Okay. Yes, very good, Srivatsan. In which of the following methods ART, fertilization, will be in vivo? Okay. So IVF ET, that is the embryo transfer, ZIFT, ICSI, AI, that is artificial insemination technique. See, infertility, that is the main thing, that where we are going for the assisted reproductive technology. So what is happening here? The problem is either can be in the female or the male. Either the men is not able to inseminate, or the sperm count may be low, or the morphology of the sperm, okay, that may not be there. So all that we can correct by using what is called the AI technique, which is the artificial insemination technique. In this, we will collect the semen from the donor, and we can, or even the husband, and we are going to introduce into the women, that is the mother, okay, to whom we wanted to plan. So either into the vagina or into the uterus itself. Okay, so this we call it as IUII, so which is very, very common. It's one of the technique where it is done in vivo. Okay, it's not IVF. IVF is done in vitro. That's why it's called in vitro fertilization. You understood? We are taking from the women, from the men, and we are going to do it in a test tube, right? That's why it's called the test tube baby. You understood, ma? Yes, the answer is last option. It is the AI technique. See here, able to follow, Srivatsa? See, infertility cases either due to inability of the male partner to inseminate the female, or a very low sperm count in the ejaculation, all could be corrected by the artificial insemination technique. What do you mean by in vivo? You are injecting it inside the female. So in this technique, the semen collected either from the husband or a healthy donor is artificially introduced either into the vagina or into the uterus of the female. Okay, so this is the method. Yes, very good. Mendel conducted the what you call the artificial pollination, cross pollination experiments using several. Option A, true breeding garden pea lines, true breeding sweet pea plants, hybrid garden pea lines, hybrid sweet pea lines. So Mendel, he carried out this hybridization experiment on the garden pea plants, okay, for many years. So he performed this various types of these breeding and he allowed the offspring to self-breed. All this is why he selected so many. He did nearly more than 7 years. Like, what is the reason? Because he want to bring what is called the true or the pure lines of breeding lines, so that they produce offspring exactly resembling that of the parents. Okay, so what is the option? Yes, children. Okay, no issues, Srivatsa, you're good. So just see what is the option. Yes, exactly, you're right, Srivatsan. Yes, see, the answer is what? It is the true breeding lines are homozygous. So exactly it will be there, and for any hybridization experiments, the parent should be used as homozygous for the gene concerned. So I already said, why do we select that pure line? Because we want exactly the resemblance right from the parent, so that the offspring is exactly the same. So that is why Mendel conducted the true breeding garden pea lines in the experiment. Okay, and the next question here is, we'll go to the next question. An organism has two unlinked genes, P and Q. Its genotype is PPQQ. How many different types of gametes will be formed? Okay, so 2, 6, 4, 8. These are the options given. So here to work out on this, children, like you need to know, yes, what is that? If genes are unlinked, okay, what is according to the theory, the laws that we studied in the Mendel, then they separate during the gamete formation. Yes, the law of segregation or not. And a hybrid PPQQ, how many gametes it can? It can have 4 types of gamete. So what are the gamete? Like they can have only one allele of a gene. So there will be 4 type of gametes. So what are they? P and Q, PQ, PQ and P and Q. Yes. So the answer is how much? It is the 4 type of gametes. Okay. Yes, students, you are able to follow. Yes, very good, Neelam, very good. We'll go to the next question. A transcription unit in DNA must have a promoter, structural gene, terminator, all of the above. So what should be the answer? So when I say transcription, it is what? It's a process of the formation of RNA, yes, from the DNA. So what all it should have? The transcription unit is actually composed of the 3, the promoter, the structural gene and the terminator. So all of the above. So in structural gene, what is going to happen? You are going to have the 2. Okay, so you can see the transcription. It's the formation of RNA, okay, from the DNA. So it has got 3 regions. It has got a promoter region, it has got the structural gene, it has got the terminator. So what is going to happen in the promoter to which that RNA polymerase will bind? It will initiate the transcription from 3' to 5'. The structural gene will code for the mRNA, and the terminator is the one which will end the process of transcription. So this concept, like, you need to know. Okay, so what is that you need to know? That there are 3 things that is present in the, mostly we study in terms of promoter, structural gene and the terminator. Okay, so this is where it is very important. Yes, very good, students. All of the above is the answer. Okay, which of the following set of options is used in translation? hnRNA, hRNA, tRNA, rRNA. The option B is mRNA, tRNA, rRNA, mRNA, tRNA, hnRNA. Option D is hnRNA, RNA, rRNA and tRNA. So what should be your option? Yes. So what should be your answer for this? So when I say this what is that, hnRNA. It is the heterogeneous nuclear RNA we are talking about. See, this hnRNA, no, it does not what you call undergo that processing. So the post transcriptional what you call the processing will convert this hRNA into the functional RNA. Okay, so then only it can participate in that protein synthesis, what we call translation. So what is going to happen? The tRNA and then the rRNA, they are the functional RNAs which is going to take part in the translation. Okay, let me tell you. See, what is translation? Like how you are going to give me a, say a statement. You wanted me to translate to Hindi or French. So I just, whatever you are giving, I am just reading it and I am replicating, or just like that I am translating whatever the content. The same way is that once when the information is come, it is going to process. So of formation of protein or polymerization of amino acid to form a polypeptide. So there is a code that it receives. So it just reads it and translate it in the formation of some proteins or polypeptide. So here all three RNAs are required for translation. The mRNA is going to provide the template. What about the tRNA? Yes, it is going to bring the amino acids. I hope if you have listened to our previous genetics lesson, we have taught you this. So it is going to bring the amino acids and it is going to read the genetic code. And what is the role of the rRNA? The rRNA is the one which is going to play a very important role in what? In the structural and catalytic role during the transcription. So all the RNA are very important in the process of translation. Yes, very good Neelambari. Yes, very good Srivatsa. Good. We'll go to the next question. Primates called Drytropithecus and Ramapithecus were existing about 1 Mia, 15 Mia, 5 Mia, 2.5 Mia. Mia is millions of years ago. Actually, they say that it is around somewhere around 15 Mia. The primates called this Drytropithecus and Ramapithecus were existing. Like you can see that they will be very hairy and walked with what you call like gorillas and chimpanzees. What about the Ramapithecus? It was like man-like, whereas the Drytropithecus was ape-like. So they say about 15 Mia primates called Drytropithecus and Ramapithecus were existing. Okay. See, this was only, see, very less hairy, it has got more hair. So they were hairy and walk like gorillas and chimpanzees. This Ramapithecus were more man-like, you can see that. Whereas Drytropithecus was ape-like. Okay. So 2 Mia, this Australopithecus probably lived in that Eastern African grasslands, and Homo erectus is 1.5 Mia. So that is how they say. Okay. So it is 15 Mia. That is 15 million years ago. So you are able to follow this concept? Yes. Select the incorrect match. So they wanted us to select the incorrect match. The examples of adaptive radiation: dolphin finches and Austrian marsupials, Hardy-Weinberg equilibrium, allele frequencies in a population are stable. Result of different adaptive radiations: lemur and spotted cuscus. Result of same adaptive radiation: bobcat and flying phalanger. See, one thing that you all need to understand here is the process of evolution of different species in a given geographical area. When you see, starting from a point and it is spreading to another regions, that is, the origin is here, it's spreading to another area, like you call that as the adaptive radiation. Like you take Darwin finches, what is happening? Like those finches, like depending on the habitat where they were, like their beak, okay, so that got really changed with it. And what about the Australian marsupial? Yes, it represents one of the best example of this adaptive radiation. So it also exhibits that phenomenon of divergent evolution also. So you can see the bobcat. So they have given all this about, so this is exactly the good example. And what about the Hardy-Weinberg equilibrium? Yes, we study the allele frequency in a population stable using this. And what about this thing? All these are, but we want the incorrect match. Look here, the bobcat is a placental mammal. But what about the flying phalanger? It's an Austrian marsupial. So they exhibit the result of different adaptive radiation, and it is a phenomenon of convergent evolution. So the odd one out is what? The bobcat and the flying phalanger. Okay. Okay children, we'll go to the next question. Mary Malone was a cook by profession. Yes. And also a carrier of a particular disease. Which test is commonly used for diagnosis of this disease? Monta test, Vidal test, Western blot and ELISA. So you can look here, Monta test we do for what? Montax test for tuberculosis, right? The Western blot, ELISA we do it for what? We do it for AIDS, right? Yes, and then for what we do the Western blot? For further confirmation of any infections, like, yeah, we can go with Western blot. But what about the Vidal? Yes, yes students, I want everybody to put in your answers. So please keep chatting. Yes, come on, interact in the class children, please. Yes, very good Srivatsav, you are given the right answer. The answer is Vidal test. So what is the disease? Yes, it is the typhoid caused by the Salmonella typhi. A classic case in medicine that was of Mary Malone, nicknamed Typhoid Mary, was a cook by profession. Like she was a silent carrier passing on the disease to so many people who came to her place or in the home where she was working as a cooking maid. So, and was a typhoid carrier who continued to spread the typhoid for several years through the food she prepared. Okay, so typhoid is we are diagnosing by the Vidal test. Yes, you are all right children. Yes, Neelambari. So Srivatsav, it's not the Western blot, it is the Vidal test and the disease is typhoid, okay, which is caused by the Salmonella typhi. Okay, here comes another interesting question which is very interesting. We'll see what is that. The dash, option 1 is the main lymphoid organ where all blood cells are, that is I think it's option 2, lymphocytes are produced. The dash, option 3 is a lobed organ located near the heart and beneath the breastbone. The option 3 is quite large at the time of birth but keeps reducing in size with age, and by the time puberty is attained, it reduces to a very small size. Both option 1 and option 3 provide microenvironments for the development and maturation of T lymphocyte. Okay, so we are seeing the options here: bone marrow including thymus, or spleen including thymus, thymus excluding bone marrow excluding spleen. We will see what is the answer. Srivatsav, no, it's answering right is not the matter. You are learning. Okay, I don't mind at all whether it is right or wrong. Please post the answer. So I will be happy to discuss more, like why did you choose, if it is going to be wrong or if it is going to be right. We are going to give a clap and then walk on. Okay, no issues. Okay, we'll go for it, so that I can explain you what is the right answer. Okay, so here the option we can see here, children always remember the primary lymphoid organs which I have already taught you, right? It is the thymus and the bone marrow. Okay, where the thymus and the bone marrow, they are the one. So the option here now we know that it is the bone marrow and thymus. So see, it is the one of the main lymphoid organ, okay, the bone marrow. So this would be the option. Now all others won't come up. Okay, so bone marrow, but here one word is coming, whether it is including or excluding. So here it is including, because from bone marrow all the hemopoietic, all the cells are produced. So it is including. So this would be the option. So the bone marrow is the main lymphoid organ where all the blood cells including lymphocytes are produced, and the answer is thymus. See, thymus is such a big one which will be located just above, that is, it will be located near your heart, but it will be beneath the breastbone, that is the sternum. Now what happens, it will be quite large at the time of birth, but it starts to regress. That means it is going to atrophy, what we call decrease in the size, as the child attains the puberty. So both the bone marrow and thymus, it's going to provide the microenvironment for development and maturation of T lymphocyte, which I already said in most of my classes that the lymphocytes goes to a process what we call pre-processing. It is thought self, non-self there in the thymus. That is why it is designated or tagged with a name as T lymphocyte. Yes ma? Very good. So the answer is what, children? Bone marrow including thymus. Okay, yeah, we'll go to the next question. A crop must be tested, okay, for at least dash growing seasons before releasing for commercialization. Options are 1, 2, 3, 4. So here, when you look into this procedure, the testing, release and commercialization, when new cultivators as they are planning up, okay, by growing those hybrids in their commercial fields. So what they are going to do? So we see that the testing, release and commercialization of new cultivators are done by growing hybrids in the research fields and recording their performance under all the factors like ideal fertilizer, the pesticides, the water supply, the irrigation and other all crop management practice. So this is followed by testing in the farmer field for 3 growing seasons at several locations of, because we want to find out with all the things with the climate and other things. So representing all the agroclimatic zones as well as with the soil, the climate, the temperature, everything. Okay, so what is the answer, ma? Yes, excellent, Srivatsav. The answer is 3. Yes, Neelambari, Tasmeem, Lakshmi, start posting your answers. Now coming to fisheries, here actually let me tell you that there are only 3 options, A, B and D, because both meant the same. Fisheries include rearing, catching and selling of fishes only, fishes and mollusks only, fishes, mollusks and crustaceans. Actually it is, when you see it, the answer should be like here all of the above, like or something like that. So but let us see, it's the same, like fishes, mollusks and crustaceans. Okay, so the answer, like when it is talking about fishery, it's actually, it is concerned with the rearing, catching and selling of fishes, the mollusks, the shellfish and the crustaceans. What are those? Crabs, prawns and all. So a large number of population is dependent on what, particularly on the aquatic regions, you know, those coastal areas, you know. This is a bread and bun. This is a bread and butter for those people, right? They'll be dependent on these only. So they dependent on these fishing and other aquatic animals like prawn, crab, lobster, edible oyster for their food. Okay, that is, they are going to sell it out and that's how they are going to have their livelihood. So yes, your answer is very good, Neelambari, Srivatsav. The answer is fishes, mollusks and crustaceans. Okay, we'll go to the next question. Yes, very interesting. The multiple ovulation embryo transfer technology, which we call MOID, is a program for herd improvement. In this method, a cow is administered hormones with dash, the option 1, like activity to induce follicular maturation and superovulation instead of 1 egg which they normally yield per cycle. Okay, they produce dash, option 2, eggs. The animal is either mated with an allied bull or artificially inseminated. The fertilized eggs at dash, option 3, cell stages are recovered, dash, option 4, and transferred to surrogate mothers. The genetic mother is available for another round of superovulation. So option here they have given FSH 8 to 32, 6 to 8 surgically, or LH 8 to 32, 6 to 8 surgically, or whether option C FSH 6 to 8, 8 to 32 non-surgically, FSH 6 to 8, 8 to 16 non-surgically. So we have to find out what is the right answer. Now to improve the cattle breeding, there are very many strategies being made, like they select the superior breed in the female, and this is how by super ovulation they are going to increase the number of eggs. So they are going to artificially inseminate so that it will produce what is called the multiple zygote embryos. So later they will be transferred to another cows where they act only like surrogate. Okay, so just for the developmental purpose. Now the mother will be ready for the next round of producing those embryos. So first thing, always children, you should understand. Yes, very good, Srivatsav. So the first thing that you need to understand here in this method, one thing I would like to tell you, see they have said here it is with the follicular maturation. So these two options will not come. See, always try to narrow down so that it will be easy for you to choose up. Now what is the next one? They have told here that which they yield normally per cycle, they produce how many? It cannot be 8 to 32. So it is usually 6 to 8 eggs. Okay, and what about the cell stage? Yes, they usually take it up the 8 or the 32 cell stage, and non-surgically is the one, it is not. So that is where this becomes the right option. Okay children, you are all able to follow? Yes. Okay, we'll go to the next question. Select the correct statement from the following. Okay, so let us see what is the correct statement. Okay, the activated sludge sediment in settlement tanks of the sewage treatment plant is a rich source of anaerobic bacteria. Okay, biogas is produced by the activity of aerobic bacteria on animal waste. Okay. Okay. Methanobacterium is an aerobic bacterium found in the rumen of cattle. Biogas, commonly called gobar gas, is a mixture of gases mainly composed of methane. Okay. Just one minute. So you can see here, like, what is the statement that is correct. Now always first to find out that the first statement they have said activated sludge sediment in settlement tanks of the sewage treatment plant is a rich source of anaerobic bacteria. No, it is the aerobic bacteria, which we call it as flocks. So this statement will not be right. What about the second one? Biogas is produced by the activity of the anaerobic bacteria, not the aerobic. What about the methanobacterium? Yes, the methanobacterium, as you see, is an aerobic bacterium found in the rumen of cattle. So methanobacterium is an anaerobic bacteria, not aerobic. So this option is wrong. But what about biogas? The biogas is commonly, which we call the gobar gas, is a mixture of gas, right? It is composed of methane, then the sulfides, yes, and then we have carbon dioxide, right? So they are all the right options. So which is the right statement? It is the fourth one, the biogas. So see, biogas, it is composed of methane, carbon dioxide, hydrogen, nitrogen, and the hydrogen sulfide, right? Of these, methane forms the 50 to 70%, 30 to 40% by the carbon dioxide, and 10 to 20% includes the other components. Yes, very good, Srivatsav, you are right. You have given the right answer. Yes, the answer is biogas. Yes, we'll go to the next question. Techniques to alter the genetic material. The RNA, DNA is known as what? We'll see one by one. See, they have given options as biomedical engineering, environmental engineering, genetic engineering, and bioprocessing engineering. Now, what is bioprocessing engineering? It is actually a field in the engineering where they deal with the design as well as the development of equipment and processes for the manufacture of products such as agriculture, chemicals, like that. So this will not be our option. What about the environmental? The environment is something like it helps in protecting, like it is a type where they design the equipments, where they help, particularly they will be studying more with the environment effects. They will make things that is going to help the society to avoid the effects or the impact of those pollution, improve the environmental quality, like that. So this will not be there. So what about the biomedical engineering? Like here it's the medical, like the biology, we are applying it with the engineering, that principle, the engineering principle, designs and all that. So that is how, where we can cater the medicine and biology for the healthcare purposes. So that is where we use. So this cannot be there. See, we have that pulse oximeter, that in every house, I think mostly we have that Omron for digital recording BP. All that is out by the biomedical engineering. So there are so many what you call novelty, I can tell you, novelty way they make. We don't still depend on the traditional methods. Okay. So the right answer is genetic engineering. So what is genetic engineering? It is known as the recombinant DNA technology. Okay. Yes, very good, Neelambari, you have given the right answer. It is a recombinant DNA technology. It is used to alter, okay, the chemistry of genetic material, that is the DNA or RNA, and it is to introduce it into the host cell so that it will change the genotype and thus the phenotype of the organism. So that is how we are using it. Okay. So I hope you know this, right? So that is how. Okay, we'll go to the next question. Use of bioreserves by multinational companies and other organizations without proper authorization from countries and people concerned without compensatory payment is known as genetic relatedness, biopiracy, biodegradation, biomagnification. So what should be the right answer? So you can see here it is the biopiracy, because some organization and even multinational companies and all know, like they try to exploit or get patent for the biological resources, okay, for of other nations without proper authorizations. So like from that concerned country, what we call that as the biopiracy. So what is biopiracy? It is the, see, suppose we have that basmati rice, the turmeric and all, no, like suppose we wanted something like from the other nation, if somebody wants to use it, they have to get that proper authorization from the concerned country, or if a person has found out, he has to get that permission from the concerned person, okay. Without that they should not carry on out. So that comes under what we call it as biopiracy, like they get patent for it, okay, it is patent, so that they can't use it without the knowledge of that particular country, because they might be traditionally having it for years together, and somebody working on it cannot get a patent and say that is mine. So it has been traditionally been years together, like how we had issues with turmeric and all. So like that, now they have made it very clear cut, you can't hold it like that. So same way, if I am taking and working on some gene and somebody cannot claim it on it, so that I have to immediately patent and file and keep it, okay. So something like that, okay. So you are able to follow. So most of the industrialized nations, they are very rich financially, but they are very poor in biodiversity as well as the traditional knowledge. So the traditional knowledge related to that bioresource is used by the developed countries by inadequate compensation, and as well as the benefit sharing to the farmers and even to the people of that specific region or country. So that is where it comes, okay. So we go to the next question. Yes students, you are giving the right answer, very good. Adaptation in an organism can be morphological, physiological, behavioral, all of these. So what could be your answer? So it is all, right? Yes, you're all right. The answer is all, because when I say adaptation, it's an attribute of the organism, morphological, physiological, behavioral, that enables the organism to survive and reproduce in its habitat. So many adaptations have evolved over long evolutionary period, and they are genetically fixed, okay. So however these may remain epigenetic, like not fixed genetically. Like for example, like we will see one example I will give, how is that adaptation. Now suppose a person, okay, they are going to live in a high altitude, okay, for generations together. Like what happens, suppose when we go, we do get what is called the mountain sickness, right? Yes, yes or no? Yes, we do get. But what is happening there in the peoples there? So they don't fall sick and all, because they have been acclimatized, what we call they are adapted, which we call it as the physiological adaptation. What we call that as the physiological adaptation, okay ma, you are able to follow. So that is called the physiological adaptation, which is quite common, okay. But what about say the example for morphological, like you can see those leopards, or even you can see the what you call the those seal and those animals that live in colder regions and all, like how they have that Allen's rule, like they have short ears and all that. So that is what we call it as the morphological adaptation. Behavioral adaptation, like suppose if it's going to be summer, like we want to have a very cold drink, or we just want to go near, sit under the AC room or a shade, all that is behavioral adaptation. So adaptation is an attribute of that organism, where morphological, physiological and behavioral, everything is what meant for ultimately what for survival. That means what, one word, homeostasis. Yes, so that should be the answer. Okay, we'll go to the next question. The detritus break down detritus into detritus into smaller particles, and this process is called fragmentation, leaching, humidification or calcification. So after this, it's a very direct question. Yes, the answer is fragmentation. Okay, we'll see how. The dead plant remains such as leaves, flower bark and dead remains of animals, including the fecal matter, constitute the detritus. So which is the raw material for decomposition. So the earthworm, they will break down those detritus into the smaller fragments so that there will be more area available for further enzymatic activity. So this process we call it as fragmentation. Okay. India has only dash of the of the world's land area. The options are 21%, 100%, 2.4% and 4.2%. Yes, students, please do post your answers. Okay. So India is a mega diversity country, I hope, with only 2.4% of the the world's land area, but it accounts for nearly around 7 to 8% of all the recorded species, around more than 45,000 species of plants and 91,000 species of animals. So that is where. So you can see India, it occupies only 2.4% of the world's land area, but it shares that global species diversity like around 8.1%. So it's one of the 12 mega diversity countries of the world. Okay. We'll go to the next question. Okay, so the next question, find the correct match. Column 1 gives Rafflesia, quinine, curcuma longa. Column 2 we have rhizome, root and bark. So what is the right option? We have to find out. See, always that Rafflesia we know it is actually used as a source of alkaloid rasparin that is used for that hypertensive drug. What about the bark? The bark is with the quinine. So the bark of the quinine is actually used for that anti-malarial drugs. What about curcuma longa? We are very much sure that they are used for the rhizome, that is the turmeric, which has got its medicinal values. Okay. Okay children, you are able to follow. So the right option is what then? It is the Rafflesia root, the quinine, which is the bark, and the curcuma longa, the turmeric, is the rhizome. Okay. We'll go to the next question. The Haryana Kisan Welfare Club is run by Ramesh Chandra Dagar, Ahmad Khan, Swaminathan, Ramdev Mishra. Yes, Srivatsa, you can leave if you have a work, no issues. Okay, so we can proceed with it. So what would be the right answer here, ma? The Haryana Kisan Welfare Club. Yes, it's something to do with integrated farming. Okay, so it is something to do with the integrated farming. So what is that going to tell us about? So this integrated organic farming is actually something where they wanted to make it a zero waste procedure, like whatever the waste they think, okay, so that comes from the farming procedure, they never waste it, and that is how they have been using it in the recycling process, and that is how they have got success at it. So to spread this information of that practice, okay, with the practice of integrated organic farming, Ramesh Chandra Dagar has created that Haryana Kisan Welfare, and he is a man who is standing there, and he is the one who has talked about this Haryana Kisan Welfare Club. Okay. We'll go to the next question. Yes, Ramesh Chandra Dagar. Yes, students, please do post your answers. So dash are useful in providing information for identification of varieties of species found in an area, whether they are called manuals, catalog, monograph or flora. So what should be the right answer? We'll see them. See, first thing that you need to understand here, when I say they are going to give information about identification of variety of species, we are talking about the manual. Now I'll tell you each one what do they say. See, when I say flora, it is going to give you the entire account detail of that particular plant you take, whether like say for example you are taking a pea plant, like what is the habitat they do live, what are the distribution. So that is confined with flora. So they will give you the index of that particular plant species in a particular area. But what about the manuals? They are very useful in giving the information of identification of variety of species found in an area. What about the monograph? It will give just about only one taxon. Okay, catalog is with like with a plant or animal catalog. Okay, with that it will again differ. So the exact answer here is what? It is the manual. Okay, so that is the right answer. Yes, children, please do post your answer. Yes, very good students. Yes, Neelambari, very good. You have given, you have said about flora, right? So flora cannot be the option, ma, because when you say flora, because you can't say flora, because flora is the one which will give you about that plant in a given area, right? It won't tell you about the variety of species and all. It talks about that particular thing in that which habitat it will be distributed and distribution. Only the manual gives you the exact result. Okay, Neelambari? Which of the following species does not have the ability to fix atmospheric nitrogen? Nostoc, Spirogyra, Azotobacter, Anabena. So this is a very direct question. I hope children you will post this answer. Yes, what is the answer, ma? Yes, the answer is nothing but it is the Spirogyra, because we all know that Anabena and Nostoc, no, they are actually they are Anabena and Nostoc, they are heterocystus blue green algae. Acha, blue green algae. What is this? Nostoc, and I am talking about the Anabena. Okay. What about the heterocyst? They are the site for the nitrogen fixation. So the Spirogyra is one of the commonest green algae. Okay. Okay, which is not having any function or role with the nitrogen fixation. Okay, so you can see here. Yes, very good, Neelambari, you have given the right answer. The nitrogen fixation is carried out by prokaryotic cells, bacteria and cyanobacteria having the NIF gene. So if NIF gene produces an enzyme, nitrogenase, which carries out nitrogen fixation. So Spirogyra is, as I said, it's a green algae that doesn't have that NIF gene, so it cannot carry the nitrogen fixation. Okay, children? Yes. So Selaginella can be regarded as evolutionary advanced pteridophyte because of rhizopore, ligule, heterospory or apospory. So what is heterospory? Is the occurrence of the two type of spores, the smaller microspore and larger macrospore. Yes, so heterospory is occurrence of what? Two type of spore. One will be small, another one is large. Yes, so heterospory has led to the evolution of seed habitat in plants. So heterospory was first evolved in Pteridophyta. Okay, so in which the megaspore was retained on the parent, the parent sporophyte, and which is considered as a precursor to sea habitat. So apart from Selaginella, even Salvinia, even this is also considered as evolutionary advanced pteridophyte, which will produce two kind of spores, the macro and the microspores. Okay, so they also produce the macro and the microspores. So that is where these two are considered to be evolutionary advanced pteridophytes. Okay, so they say that heterospory is the one which leads to seed habits in the plants. Okay, yes, very good, you have given the right answer, ma. Adult annelids have symmetry similar to that of adult coelenterate, most of the sponges, adult echinodermates and chordates. So we will see the right answer. See, when you see adult annelids, they have that that symmetry, which is bilateral symmetry, that we are aware of it. Now see, animals like annelids, arthropods, chordates, larvae of echinoderms, etc., where the body can be actually divided into the identical right and left halves. Okay, so they can be divided into right and left halves. Okay, in only one plane, that is exhibiting what we call it as the bilateral symmetry. So the answer of this annelid is bilateral symmetry. But what is happening when any plane passing through the central axis, okay, of that will divide the organism into two identical halves, that is the radial symmetry. Like echinodermata and all, starfish and all, what they have? They have the radial symmetry there, right? The adult echinodermata has what? Radial symmetry. Okay, so this will not be there. Most of the sponges, they have asymmetrical. So what will be the adult coelentrates? So adult coelentrates also, they have radial symmetry. So what is the option? It is the chordate. So the answer is chordate. So sponges are mostly asymmetrical, that is any plane that passes through the center does not divide them into two equal halves. So that is where they are asymmetrical. Yes, we'll go to the next one. Members of which phylum lack, okay, members of which phylum lack a well-defined circulatory system and a respiratory system? Aschelminthes, Annelida, Arthropoda and Hemichordata. So when you look into this option, the circulatory system actually was first seen in the Annelida. See, in Annelida we have that closed type of circulatory system. But what about others like Arthropoda and Hemichordata? They will have the open type of circulatory system. Okay, they have that open type of circulatory system. So it is the Aschelminthes, as you see. So it is the one which does not have that is the roundworms, no? They don't have the circulatory system or the respiratory system. Okay, ma? Understood? So we'll go to the next question, the 30th question. Yes, it's very interesting question. Select the correct statement. A bud is present in the axil of petiole in both simple and compound leaf. You have to find the correct statement. Please remember that. Swollen leaf base occurs in Solanaceae. Stem tendril occurs in P. Phyllocaid is expanded petiole which becomes green and photosynthetic. So what should be your answer? Yes, students, please do post your answers. Yes, the answer here is when you see the bud. So you know that the bud is actually, it is going to be present in the axil of the petiole in both simple and compound leaf, but it is not present in the compound leaf, right? So the answer is this. This is the right statement. What about the swollen leaf base? It is actually the swollen leaf base, we will find it in the Fabaceae family. So the answer here is Fabaceae. Okay, and what about the stem tendril? No, it is not the stem tendril, it is actually the leaf tendril that will occur in the pea plant. And what about the phyllocaid? We are aware of it, that the phyllocaid is nothing but it is the expanded stem, the photosynthetic stem. The phylloid is the one which is the expanded green petiole that is going to take part in the photosynthesis. Okay, so this clarification, so this will also not be the answer. So option A is the answer. Yes, very good. Which one of the following statement pertaining to plant structure is correct? Cork lacks stomata but lenticels carry out transpiration. Passage cells help in transfer of food from cortex to phloem. Sieve tubes possess cytoplasm and nuclei. What about the shoot apical meristem has a vascular tissue? Now when you look into it, we are going to see which one of the following statement is correct. So when you see the correct statement, see, when I say cork, it's actually a dead tissue, okay, that lacks stomata but lenticels person carries the transpiration, also the carries the gas exchange. Okay, now the passage cells, when you see, so that is the right statement. So cork doesn't have stomata, right, but lenticels which you see there, that is the one which is going to perform the role there. What about the passage cell? They are actually present in the endodermis and it is going to regulate the unidirectional flow of water and minerals in roots. Okay, so that is not the answer. What about the sieve tubes? They don't have cytoplasm or nucleus because the exact thing is done by the companion cell, because companion cell regulates its functioning. What about the shoot apex? It consists of meristematic tissue only. It does not the shoot apical meristem, it does not have what you call the vascular tissue. So that should be the answer. Yes, students, can we move to the next question? Which of the following act as a supporting framework? Okay, which of the one will act like a supporting framework for epithelium? So the answer is very quite straightforward. Areolar tissue, adipose tissue, dense connective tissue and cartilage. So when you look for the supporting, yes, it comes the areolar tissue. See, the loose connective tissue, no, it has got those cells and fibers. So they will be arranged in a semi-fluid ground substance. Okay, for example, the areolar tissue that is present beneath the skin. So that is going to act like the framework for epithelium. So what all the constituents? It will have fibroblast, macrophages. Okay, but what about adipose tissue? It's a modified areolar tissue which is immediately beneath the skin, which is going to store the fat. Okay, so the excess of nutrients that are not used immediately are converted into fat and they are stored in the adipose tissue. So the supporting framework is what? It is the areolar tissue. Okay, so I hope you understood is why we chose the areolar tissue, right children? Yes, yes, very good. Yes, very good Neelambari, you have given the right answer. In which of the following would you expect to find glyoxysomes? Okay, endosperm of wheat, endosperm of castor, endosperm of gram, leaf cells. See, first you should understand what is glyoxysomes. They are nothing but they are the micro bodies, ma. Okay, so these micro bodies are not only they are rich in plant cells like castor seeds. Okay, they take part in the beta oxidation of fat. Okay, so they possess one important enzyme, catalase, that is very important of this glyoxysomes. So we see here the glyoxysomes are present in the fat rich seed. So it is going to carry out breakdown of fat into carbohydrates by glycoxylate cycle during the seed germination. Okay, to provide energy for the growing embryo. So it also occurs in other fatty seeds like ground, mustard and all that. So where is the option? It is the endosperm of castor. As we'll see the next one, which of the following is not correctly matched with its category? Insulin, carbohydrate, GLUT4. GLUT4 means glucose transporter. Okay, GLUT4 protein, lecithin phospholipids, cholesterol lipid. So here which is not correctly matched? So see, insulin means what? It's a hormone, right? It's a hormone, it's a protein hormone having 51 amino acids. It's not a carbohydrate, right? So it's a protein hormone, and insulin is actually produced by the A cells of the islets of Langerhans in the pancreas. Okay, so GLUT4 is a protein. It's actually a transporter which helps in the presence of insulin. It is actually present in the muscle and all, going to help in transport of what? It is going to help in the transport of the glucose. The lecithin with phospholipids, cholesterol with lipid, all are rightly matched. So the wrong option is insulin and carbohydrates. Okay, yes, we'll go to the next one. Find out the number of chromosomes. Find out the number of chromosome and the amount of DNA in a spore. Okay, mother cell of bryophytes in G2 stage. If the spore contains 5 chromosomes and 5 picogram of DNA, 5 chromosome 5 picogram DNA, 10 chromosome 20 picogram DNA, 10 chromosome 10 picogram DNA, 20 chromosomes 20 picogram DNA. So it is given that the spore of the bryophytes, okay, it contains 5 chromosomes and 5 picograms of DNA. Now spore is what? It is haploid. Okay, the spore is what? It is haploid. The spore mother cell is diploid. We know that. Okay, so due to this, so okay, you should know that the spore, I said it's a haploid. What about the mother cell? It is diploid. Now due to this, the spore mother cell will have double the amount of chromosome. So during the G2 cell phase, what is going to happen? It is going to double up. So what is going to happen? The spore mother cell of the bryophyte will be 10 chromosome, and what will happen to the DNA? It will be 20. So the answer is 10 chromosome and 20 picogram of DNA. So children, you are able to follow. The spore mother cell is diploid. What about the spores? They are haploid. The mother cell is diploid. So if a spore has 5 chromosome, 5 picogram of DNA, then how much it will happen? So during the G2 phase, there will be doubling up. They'll remain unchanged. So since the DNA replication occurs during the S phase, but the chromosome number remains unchanged. So chromosome in G2 is how much? Is 10, and the quantity of DNA is 20 picogram. Root pressure is maximum when the options are transpiration is high, okay, the transpiration is high and absorption is very low, yeah, or the transpiration is very low and absorption is very high. Transpiration and absorption both are high, transpiration and absorption both are low. So what should be your option? So when the root pressure, as we are talking about, yes, see, the transpiration will be very low. What happens when the pressure is going to be very maximum? What will happen to the transpiration? It will become very less, and absorption will become very high. So that will be the option. So root pressure is a pressure that will develop in the xylem, the trachary elements of the xylem. Yes, very good. So that will develop in the trachary elements of the xylem due to accumulation of water. So it occurs due to the active absorption. You can see the active absorption of water, and it will be maximum. So the rate of absorption will be what? It will be very, very high, and at the same time the transpiration will become very low. Select the incorrect pair from the following. Photolysis of water, manganese chlorine, cation anion balance, potassium chloride, redox element. Yes, redox element. A redox element, it is iron, copper, phloem, transport boron and copper. So which is not rightly given here. So as you see here, it is the phloem transport because that is carried out by boron, not copper, it is potassium. Because boron helps in translocating the solute. So it is very much important for regulating the pressure gradient of the sieve tube element of phloem. So that is the right answer. Okay. Yes, students, please do post your answers. In light reaction, the assimilatory paths produced are ATP NADH2, ATP NADPH2 plus H plus, ATP FADH2 NADP. So what happens in the light reaction? Yes, we are aware of it. In the light reaction, the light energy whatever from the sun is trapped, and what is happening? There is a reaction where we have the photosystems 1 and 2. With that, with the help of that, the ATP NADPH2 will be produced. Then these energies will be used in the dark reaction, particularly the Calvin cycle and all, where it helps in the synthesis of glucose. So thus known as the assimilatory path. So the answer is what? ATP NADPH2 H plus. Okay, so that is the right answer. Yes, very good, Neelambari, you have given the right answer. Carbon dioxide acceptor and carboxylating enzyme in mesophyll cell of C4 plants are respectively PEP Rubisco, RUB pepcase, PEP pepcase. Last option is 3-PGA and Rubisco. So when you look into the C4 plants which has got that characteristic Kranz anatomy, the carbon dioxide acceptor there, it is the PEP, phosphoenol pyruvate, and it is a phosphoenol pyruvate carboxylase. That is the one which is going to help. So you can see here in this C4 plants which has got the Kranz anatomy, that means it has got the mesophyll cell and it has the bundle sheath cell, where the PEP will be converted by the PEP carboxylase, that is the initial one to accept, and at the same time it is the one which will convert into the first carbon dioxide product, that is the 4C compound of oxaloacetic acid. Then it is converted to malic acid, which is then transported inside the bundle sheath. Okay, that in turn will enter the Calvin cycle. So the exact answer is what? It is the PEP and pepcase. So the C4 plants has got that Kranz anatomy, okay, in which the mesophyll chloroplast and the bundle sheath chloroplast, they will differ. So the reaction which will occur in the mesophyll, they are catalyzed particularly by the PEP carboxylase enzyme, and the initial product acceptor is 3-carbon compound. Okay, we'll go to the next question. In aerobic respiration, there is complete oxidation. Okay, there is complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, releasing dash molecules of carbon dioxide. The options are 2, 3, 4 and 6. So the exact answer is what? Yes, it is the 3. Okay, it's because pyruvate is a 3-carbon compound. So once when there is a complete oxidation, it is going to produce 3 molecules of carbon dioxide, and that is the one which is going to get converted to the acetyl CoA and in turn enters into the Krebs cycle. Okay, so pyruvate is a 3-carbon compound where there is complete oxidation. So what is happening? There is 3 molecules of carbon dioxide, of which 1 molecule in the link and 2 will be released during the Krebs cycle. How many of the following tissues given below are formed by dedifferentiation? Okay, dedifferentiation, like once again it is regaining the what you call the capability to divide. Okay, so intrafascicular cambium, interfascicular cambium, cork cambium, procambium, phelloderm. 3, 2, 4 and 5. So what should be your answers, children? So I said already that it is something dedifferentiation. It is a process by which the permanent tissue like parenchyma regions, they have the power of division and they become meristematic again. Such meristematic is called secondary meristem. So what is the one they have given? Interfascicular cambium, cork cambium, yes, cambium. So 2 is the right answer they have given here. Okay. Yes, Neelambari, Tasmin, post your answers. Which of the following is not correctly matched? Muscularis mucosa, it is the outermost layer and it is made up of thin mesothelium with some connective tissues. B option, hepatic lobules. These are the structural and functional unit of liver containing hepatic cells arranged in the form of cords. Parietal cells, these secrete HCl intrinsic factor in stomach. Diarrhea, the abnormal frequency of bowel movement and increased liquidity of the fecal discharge. So which is not correctly matched. So hepatic lobules, yes, it's the structural and functional unit of the liver. Parietal cells that secrete HCl, an intrinsic factor in the stomach, intrinsic factor needed for vitamin B12 absorption. What about diarrhea? Yes, abnormal frequency of bowel movement, so there is more of stool discharge. Yes. So this one is wrong because the muscularis mucosa is the innermost layer, not the outer one. So the serosa is the outermost layer, okay, with some connective tissue. Muscularis is actually the smooth muscle that is arranged into the what you call the circular and outer longitudinal layer. So the innermost one will be the mucosa, then that is followed by submucosa, then we have what is called the then we have what is called the muscular layer, we say muscular layer, and then we have the serosa. This is from inside to outside. Okay, so an oblique muscle layer may be present in some region, particularly stomach. Okay children, we'll go to the next question. A specialized center present in the brain called dash, okay, is responsible for the regulation. Another center present in the pons. So another center, yes, very good ma. Another center present in the pons region of the brain called dash can moderate the functions of option A. Neural signal from this center, option C, the duration of inspiration and thereby alter the respiratory rate. Option D is situated adjacent to the rhythm center, which is highly sensitive to option E. Children, one thing you should understand. So they said there is a specialized region in the medulla, okay, that is responsible for the regulation. So when it is come A, I want you to say the answer is the respiratory rhythm center. See, all this is not the answer. It is a respiratory rhythm center, which we call it as dorsal respiratory group of neuron. They are responsible. There is another center in the pons, that means it is the pneumotaxic center. Yeah, both are coming correct. Now what is the one? It can dash the function of A. What the function? The DRG, whatever function. The neural signal from this center can dash the inspiration. It is actually reduce. It is not increase. So it is going to reduce the duration of inspiration. Rather, we use in our medical terminology, limit the duration of inspiration and thereby alter the respiratory rate. Okay. So dash is something adjacent. It is the chemosensitive area and they are very highly sensitive to carbon dioxide and hydrogen. Yes, very good Neelambari, good. The last question of the day. Serum is having all of the following except ions, glucose, clotting factors, amino acids. So serum is what? Very simple. We do teach in our college saying that serum is what? It is plasma minus clotting factors. That's it. So plasma minus clotting. You are taking a blood not added with any anticoagulant and I am just keeping it aside. And after some time when I take and centrifuge, I am going to collect the serum because plasma minus. So it will have all the constituents same like the plasma except the what? The clotting factor, because that has been used and made as a clot. So blood without clotting factor is called serum, while blood without RBC and certain plasma protein is lymph. Okay, the answer is clotting factors. Okay, so with this I think I am ending up the part 1 session of this mock test 6. Okay, I think today it was very nice. Many of them interacted and gave answers. It was very nice. See you soon in the next session. Thank you students. Bye.